Now that you understand genetics and the processes of inheritance, we are going to look at the origin of species by natural selection, or evolution. This is Genes Within Populations, Chapter 20. Scientists have been trying to explain the changes that they saw in living things and in populations for quite some time before Charles Darwin proposed his theory of evolution. He said that species change over time, and that new species were the result of the total of all of these changes. When Darwin published his book, Origin of the Species by Means of Natural Selection, his theory of evolution was immediately challenged. Darwin's primary rival was Jean-Baptiste Lamarck and his theory of inheritance of acquired characteristics. According to Lamarck, changes that an organism accrued during their lifetime could be passed on to their offspring. Now this sounds similar to Darwin, except Darwin was talking about genetic changes. Lamarck was talking about actual physical changes that occurred to the parent during their lifetime. His example was giraffes. In his ideas, giraffes originally had short necks. They stretched their neck to reach leaves that were higher in the trees. They passed this slightly stretched neck on to their offspring, who in turn stretched their longer necks to reach even higher leaves, and so on until we have the long-necked animals of modern times. So where Darwin said you had to have a genetic change, Lamarck said a parent changed and they automatically could pass that to their offspring. So according to Lamarck, if you join the track team and over time your running speed increases, you will pass that improved running speed on to your children automatically. When Darwin spoke of evolution, he talked of descent with modification. We would say evolution is a change in a population's genetic makeup over time. This would suggest that all modern species have descended from earlier ancestral species. The process behind evolution is the idea of natural selection. Natural selection means that nature selects those individuals that are the best fit for the environment at the time. If we go back to the giraffe idea, natural selection will say that within a population you will have a variety of a particular trait. So if we're going to talk about necks, we'll have those individuals that have short necks, those that have average necks, and those that have longer necks. If the longer neck gene allows that organism to reach more food, then it will be healthier and it will make more babies. Those babies, in turn, will be healthier and make more babies, and so on, until eventually the entire population will have a long neck. That population, or that species, will have evolved. Keep in mind, evolution does not always lead to increasing complexity, but it does always lead to a better fit with the environment. The process of evolution begins with the DNA, or the gene. If the gene changes, then you have a mutation. Remember, mutations are neither positive nor negative. It's the trait that's positive or negative. If the trait is non-beneficial or negative, it will die, either because the organism that has the trait dies, or it's not chosen as a mate, so it can't pass that trait on. If the trait is beneficial, then it is passed to offspring, who in turn pass it to their offspring. Over time, natural selection occurs. That means nature selects this new version over the old version. Over time, again, we have microevolution. Microevolution is the change between the original organism and the naturally selected organism. If, again, over time, those organisms no longer mate, either because they physically can't or they won't, then you have macroevolution, or a new species. Genetic variation within the individuals is the raw material for natural selection, but there are 3 criteria. Number 1, the variation must exist among the individuals. Number 2, the variation among the individuals must result in differences in the number of organisms that survive to the next generation, meaning that the babies with the new trait must survive in larger numbers than the babies without the new trait. And number 3, that variation must be genetically inherited. If that trait or that gene cannot be passed on, you cannot have evolution. Remember, evolution is the result in a change in the composition of a population. Individuals do not evolve. Natural selection and evolution are not the same thing. People get them confused. Natural selection is the process, evolution is the result. Evolution, driven by natural selection, is going to create a population that is better adapted or a better fit to the environment. That's what the whole process is about, is to create an individual, a population, that is the best fit possible for the physical environment. My example is pocket mice that live in New Mexico. Pocket mice come in 2 colors, black and tan. Originally they were tan, the same color as the sand they lived on. This was the best fit because they weren't seen by predators. Part of the population broke off and moved to a nearby lava field. Unfortunately, their tan color stood out on the black rock and they were easily seen by predators. Those individuals that had the gene for darker fur were able to survive, and over time their fur changed from tan to black, making them very difficult to see by the predators. The difference between the tan and the black colors is microevolution. I'll discuss my second example, which are the peppered moths of England, in class. I've said several times that natural selection and evolution lead to an organism and a population that's the best fit for its environment. But exactly how is fitness measured? Fitness is the most fit phenotype that produces the greatest number of offspring. Remember, I've told you several times that an organism measures its success by the number of babies it leaves behind. If we're looking at evolutionary fitness, we're going to look at several different parts of that success. First, of course, you have survival. You have to be better at getting food and better at not becoming food. So survival is important in fitness. Of course, if you're going to be a fit individual, you have to be successful at attracting mates, because you have to have more mates in order to produce more babies. And then, ideally, you want to have more babies each time you mate than others. By doing this, then you leave more genes than your rivals. If those genes carry that new gene, that naturally selected gene, then you're going to put more of your new trait out into the environment, and it's going to make you and your babies more fit. Your fitness is greater than those individuals that don't have your new trait. Over time, the genotype and its responding phenotype that has the greatest fitness will increase in frequency until it becomes the one that everybody centers around. There are 4 agents of evolutionary change. The first is mutation. That's what we've been talking about. It's the ultimate source of genetic variation, and it makes evolution possible. However, a gene mutates only once every 100,000 replications. Because of that, the effect of mutation on evolutionary change is generally low compared to other processes. The second of those processes is gene flow, which is the movement of alleles from one population to another. You can also call it immigration, which was the movement of alleles into the population, and emigration, which is the movement out of the population. This can happen in a variety of ways. For example, an animal can physically move into a new population. Pollen or seeds can blow into an area on the wind. Or we could have matings between individuals whose populations run into each other. If that animal or that plant has a characteristic that's new to the population, and it can adapt to its new environment, and it is able to be passed on to new offspring, then it might be a source of evolutionary change. The third agent of evolutionary change is non-random mating. In plants, this is self-fertilization. In animals, it's sexual selection. In most species, the female chooses her mate, and if she's doing this by non-random mating, she's going to choose him based on a trait that she prefers. An example of this would be feather color in birds. Our last agent of change is the idea of genetic drift. Genetic drift is the result of a catastrophic event that the population is not in control of. This could be disease or a flood. In this case, the alleles that survive aren't necessarily more fit, they're just more lucky. The impact of genetic drift is dependent on the population size. If the population is small, genetic drift can have a huge impact on the population. If the population size is large, it might be big enough that it can actually absorb genetic drift, and the evolutionary change is very slight. Natural selection is that process that increases the frequency of beneficial alleles. In turn, those beneficial alleles will enhance organism survival and increase the number of surviving offspring. But natural selection is not a guaranteed process. It can be modified by some of the other processes that we've talked about. The first is mutation. Even though mutation starts the process of natural selection, it can actually alter it. If, for example, a beneficial allele mutates into a non-beneficial allele, and that mutation happens fast enough and often enough, it can actually force the population to go in a different direction than natural selection would want. In nature, mutation rates are rarely high enough for this to actually happen. Then we have genetic drift. Since drift is random, it can decrease the frequency of a favored allele very quickly. Worst case, it can actually cause that favored allele to disappear. For example, you have a population of wading birds, and all the birds with the favored trait of longer legs are washed away by a flash flood. For that population, that favored allele is gone. Finally, we have gene flow or migration. Migration can be either constructive or constraining. If you have constructive migration, individuals with a beneficial mutation will move into a new population. So, for example, we have a population of mice. In this population, large ears are favored because they allow the animal to better hear a stalking predator. In a constructive situation, animals with large ears will migrate into the population that already has small-eared mice, and this will bring the favored allele to them. In a constraining situation, individuals with an inferior allele will move into the population. So in our example, animals with small ears will migrate into a population of large-eared mice, reintroducing a less favored allele back into the population. I want to go back to the idea of genetic drift and look at the bottleneck and founder effects. A catastrophic event has happened and most of the individuals in the population have died. The survivors have an excess of resources, so they will be reproductively very successful. The problem with this is that there has been extensive loss of species genetic diversity, meaning most of the genes in that population have disappeared because of that loss of life. An even more extreme effect is the founder effect. In this case, the catastrophic event was so extensive that we're down to just a few individuals, possibly even a single female. The new population that will come from these individuals, or this single female, will be almost genetically identical because only a small fraction of the total genetic variation of the original population has actually survived. My example of the bottleneck effect is the bearded vulture. Its habitat is in the Alps, and it was hunted to extinction in the wild. People there thought it was eating their goats, their lambs, and their small children. There were some animals in different zoos. So in the 1970s, German biologists started a reintroduction program. The program was successful and the bird was returned to the Alps. The problem is that the original population that now all these birds are related to is limited to the 120 animals that were found in the zoos at the time. Cheetahs are my example of the founder effect. About 10,000 years ago, there was a drastic change in the climate, and the cheetah population was decimated. They believe that at one point the population was reduced to a single female and 2 males. Because of this, the cheetahs of today are as genetically alike as identical twins. As I've been talking about natural selection, I've been talking about single genes and single traits. But there are many traits that are affected by more than one gene. So when natural selection is working on a particular trait, it's actually working on more than one gene. How the overall population is changed then is going to depend on which of the genes are favored. This is called multiple gene natural selection. And there are 3 types. The first is disruptive. It pays to be very different. The second is directional. There can be only one. And the third is stabilizing. It pays to be average. And I will look at each one of these in the upcoming slides. Diversifying or disruptive natural selection eliminates the average individuals. Instead, the individuals at both ends of the normal range are favored. That's why I said it pays to be very different. If you look at the graph, the individuals with the average body size are going to be eliminated, and so we'll have either large individuals or small individuals. If we look at the natural world, we're going to look at the African black-bellied seed cracker finch. Food resources for these birds are either large seeds with hard shells or small seeds with soft shells. Disruptive selection favors birds whose beak suits one seed size or another. Directional natural selection is change that favors individuals with traits at one end of the normal range. In other words, individuals at the other end of the normal range are eliminated, and the entire population then is pushed in one direction. So if you look at my graph, you'll see that the individuals with body size around 100 grams or larger are favored, and all of the others are eliminated. Over time, the population begins to shift toward that 100-gram size. In a real-world example, I'm going to fudge a little bit, and I'm going to look at artificial natural selection, or the breeding of greyhounds. Man started breeding greyhounds in 3 BC. Early breeders were interested in creating a dog that had the greatest possible speed. They carefully selected from a group of hounds those who ran the fastest. From their offspring, the breeders again selected those dogs who ran the fastest. By continuing this selection for those dogs who ran faster than the rest of the other hound dog population, they produced a dog who can run up to 60 kilometers or 40 miles per hour. Our last type of natural selection is stabilizing natural selection. In this case, individuals at both ends of the normal range are eliminated, and the average individuals are favored. So what happens are the individuals at either end, at the extremes, are eliminated. If you look at the graph, we're looking at body size. Anything between, say, 60 and 80 grams is considered average. Those individuals that are larger or smaller than that are eliminated until eventually the entire population is very close to that preferred size. If we look at a real-world example, again I'm going to fudge, and I'm going to look at that genetic breeding of the husky. Huskies were bred to pull sleds and equipment through heavy snow. These dogs have strong chest and leg muscles, allowing it to move through the dense snow easily. If the husky had heavy muscles, it would sink deep into the snow, so they would end up moving very slow or potentially get stuck. If the husky had lighter muscles, it wouldn't be strong enough to pull the sleds, so it wouldn't have the value as a working dog that they desired. Stabilizing selection has chosen the norm to be the average size for the muscles to make the dog the most efficient working dog possible. Everything in the world has a limitation. Natural selection is no different. The first of these limitations is lack of genetic variation. You have to have variation within a population in order to have natural selection. If everybody is the same, then nothing can change. So no genetic variation means no natural selection, which in turn over time means no evolution. The second limitation is pleiotropy. In this case, an individual allele has more than one effect on the creation of a phenotype. We talked about this previously when we talked about sickle cell anemia and malaria. When we're talking about limitations of natural selection, we're looking at how these multiple effects tend to set limits on how much a phenotype can be changed, like a check and balance system. For example, we breed chickens to lay more eggs per laying. That means a larger clutch size. But clutch size is tied to eggshell thickness. The more eggs that are laid, the thinner the shells are. Eventually, if clutch size is increased enough, the shells become so thin they break when the hen sits on them to warm them. So in this example, clutch size and shell thickness are related and act as a check and balance system. Our last limitation is the idea of epistasis. In this case, An allele for one gene may have many different effects depending on the presence of other alleles. Say, for example, you have a gene for skin color. If this gene, we'll call it gene A, is by itself, your skin is a natural color. If, however, you have this gene A and you have a different gene, gene B, and they appear together, your skin is not a normal color, but it's blue. That would be epistasis. Now, if we're talking about limits of natural selection, it would be as if blue is a non-beneficial trait. Therefore, you would not be chosen as a mate, and this would be a limit to natural selection in adding the blue skin trait to your population. Scientists like to be able to predict the possibility of a particular gene showing up in natural selection or evolution. In order to do that, first you need to know what the gene pool for that population is. And the gene pool is all of the alleles for all of the DNA for all of the individuals. Once you know what the possibilities are, then you can calculate the possibility of a particular allele being the one that forces natural selection. Each allele has its own frequency in a population, and that frequency can be calculated. The greater the frequency of an allele, the greater the possibility that it will be the gene that drives natural selection for that population. The calculation for this is the Hardy-Weinberg equation, and we'll be looking at that next. All the genes in a population don't evolve all the time. We talked about the factors that can stop natural selection: pleiotropy, lack of genetic frequency, and epistasis. One way to determine if a population is evolving for a particular gene is to do a calculation to determine what the genetic makeup of the population would be if it wasn't evolving for that particular gene. Then compare it to actual population data. If the data matches, the population is not evolving. If the data doesn't match, then the population is evolving, and you can start designing experiments to determine what process is behind the evolution of that particular gene. The Hardy-Weinberg principle can be used to determine if a population is not evolving. If a population is said to be in Hardy-Weinberg equilibrium, it isn't evolving. When a population is in Hardy-Weinberg equilibrium, genetic frequencies are not changing. But this has several requirements. First, no mutation is taking place. All the genes stay the same. Number two, no genes are transferred to or from other sources. This means no gene flow, no immigration, no emigration. Number three, mates are chosen randomly. There is no non-random mating, meaning mates cannot be chosen by color or size or any other selection criteria. Number four, the population size must be very large. This is in case genetic drift happens. If genetic drift happens, it cannot affect the frequency of the alleles. And number five, no natural selection is occurring. This means that all traits must have equal benefit for survival and equal benefit for number of offspring surviving. What this really means is that Hardy-Weinberg is hypothetical. All populations do evolve. It will be used as a comparison in our calculations. We're now going to start the Hardy-Weinberg calculations. The first thing you need to know is that P stands for the frequency of the dominant allele, and Q stands for the frequency of the recessive allele. You also need to know that P plus Q equals 1. And this is because if you add all of the dominant alleles in a population to all of the recessive alleles in a population, you'll get 100%. P squared plus 2pq plus q squared equals 1 is the Hardy-Weinberg equation, and you're going to need to memorize that. In this equation, p squared is homozygous dominant, q squared is homozygous recessive, and pq is heterozygous dominant. I'll explain where the 2 comes in the next slide. I want to show you where the variables within the Hardy-Weinberg equation comes from. So if you take a look at our box, this would be like a monohybrid Punnett square. They've just changed the individual box shapes on the inside. The upper left corner represents the dominant allele. Together they make up p squared because you're getting a capital A from mom and a capital A from dad. The bottom right corner represents the recessive allele because you're getting a little a from mom and a little a from dad. Together they make up q squared. The other 2 squares represent a capital A from one parent and a little a from another parent. That makes pq. And because we have 2 boxes, we have 2pq. That's where that 2 in the equation comes from. Before we get too far into the Hardy-Weinberg calculations, you need to know the difference between allele and gene frequency. When you're asked the frequency of an allele, you're being asked the number of dominant or recessive genes in a population. If you're asked for genotype or often trait frequency, then you're being asked for p squared, q squared, or pq. If you look at the examples, both ask the same question. What is Q for this population? The difference is the information provided. In question number 1, we're told that the recessive allele is 10%. Because we have the word allele, we're looking for Q. All populations are considered to be 100 unless another number is given. So 10% of 100 is 0.10, so Q is 0.10 for this population. In question number 2, we're told that 16% have the recessive trait. When we're looking at trait, we're looking at Q squared. But we don't want Q squared, we want Q. So what we're going to do is change 16% to 0.16 and then square root that to get 0.4. So Q for this population is 0.4. So let's try one. We have a population of fruit flies that have 1,000 individuals. 640 of them have red eyes, while the remainder have sepia eyes, which is that kind of brown color. The sepia eye trait is recessive. How many individuals would you expect to be homozygous for red eye color? So we're asking about individuals and we're asking about eye color, so we're looking at a trait, which means that we are looking for a square. We know that red eyes are dominant because it says sepia is recessive, so we are looking for p squared. To do a Hardy-Weinberg, you have to work in reverse. Your first step is to always calculate q squared. So we've got 1,000 individuals. We're going to subtract 640 that have the red eyes to get 360 flies with sepia eyes. That's going to be our q squared. 360 divided by 1,000 gives us 0.36. Step number 2, we're going to take the square root of that 0.36 to get q. That number is going to be 0.6. Now we got to go back. Remember I told you p plus q equals 1 because all of the dominant plus all of the recessive allele equals 100%. So in step 3 we're going to figure out what p is. we're going to take 1 minus 0.6, which is the Q that you just calculated, to get 0.4. So we know that 0.4 is P. But remember, we were asked for a trait, so we need P squared. So we're going to take 0.4 and square it to get 0.16. But you can't have 0.16 of an individual. The question asked how many individuals. So we're going to take 0.16 and times it to 1,000 to get our answer of 160 red-eyed flies. We'll be doing a lot more of these example problems in class, but if you'd like to get some extra help before class starts, you'll find a wonderful example on Bozeman biology. The teacher's name is Paul Anderson, and he's very good at explaining Hardy-Weinberg problems and just about anything that's AP biology related. I encourage you to go to his site. So you want to go to YouTube, type in Bozeman biology, and that will get you everything, or Bozeman biology Hardy-Weinberg if you're looking for examples specific to what we're doing now. And this is the end of Chapter 20, Evolution.